Sympatric speciation is another way that cladogenesis can occur. So again, I want to remind you that cladogenesis is when you have an ancestral population that is going to split into two or more new species. And so when we were talking about allopatric speciation, we said that that ancestral population was going to somehow get split. And in the case of allopatric speciation, that was a geographic barrier. And so that's why allopatric means different land or opposite land. Whereas sympatric speciation means same land. So in other words, it's not caused by geographic separation. So it will lead species to diverge even though there are no physical barriers to interbreeding. So we don't have a mountain range, we don't have an isthmus, we don't have them separated by islands. Something else is going to have to cause a block in gene flow. So it's still a block in gene flow, but it's not a geographic block. Instead, it's going to be differences in the genetics or adaptations to local environments or sexual selection that are going to block the gene flow even though organisms are living in the same area. So the first way that we can get populations splitting, right, so getting a block in gene flow even though they are living in the same area, is polyploidy. So ploidy refers to chromosome counts. So I want to start out by refreshing your memory about, for example, the term diploid, haploid. So haploid means a cell that has one set of chromosomes. Diploid is a cell that has two sets of chromosomes. And then you can have triploid or tetraploid organisms. So that would be 3 sets or 4 sets. So polyploidy is when you have any more than 2 sets. So triploid and tetraploid are both examples of polyploidy. These occur through non-disjunction. This is a failure to separate during meiosis. So again, tapping into your memory of cell division from previous classes. So the chromosomes normally split apart during meiosis. If they fail to split apart, then you can end up with extra sets of chromosomes. So sometimes this happens within an organism, and so it creates self-polyploidy. That's what autopolyploidy means. So they end up with an extra set because they carried out non-disjunction. Sorry, just modify my definition here. It can be a failure to separate during meiosis or mitosis. In the case of reproduction, it's during meiosis. So alloploids are caused by interspecies breeding. So this is when you have chromosomes from 2 or more different species, right? So you crossed 2 different species. Now you have a chromosome pair where one comes from one species and the other chromosome comes from the other. So that would be alloploidy. And so, especially in plants, you can get allopolyploidy, where they end up with being triploid or tetraploid and having chromosomes from multiple species. And when that happens, it can create reproductive isolation, which, remember, that reproductive isolation blocks gene flow. In plants, this is particularly common. You don't see it very much in animals. The reason is because plants tolerate abnormal chromosome counts much more than animals. Animals mostly don't tolerate abnormal chromosome counts. They won't be viable. But in plants, for whatever reason, they're able to have polyploidy more. And so one example that we can look at is a cross between 2 types of plants. So if you cross one type with the other type, the blue and the red, you get an interspecies cross. This is the alloploid. And then if the alloploid becomes an allopolyploid, so now this is a tetraploid individual, it actually becomes a new species. And so the reason why we consider it a new species is because it can cross with itself. So its scientific name is Galeopsis tetrahit. If you cross Galeopsis tetrahit with another Galeopsis tetrahit, you get viable future generations. So now they're able to mate with each other and perpetuate a new species, but they can't mate back with the original species. So remember, these are the original species. If I try and mate the hybrid with this original species, I get sterile offspring. And if I try and mate the hybrid with this original species, I also get sterile offspring. And the reason is because the chromosomes don't match. So they have this double set here and a second set and a third set here. So these are actually triploid. And so that would, and the triploids have more trouble being viable, or rather carrying out meiosis and making viable fertile offspring. And so again, sterile, fertile, and so this has generated a new fertile species that can't mate, right? So blocks gene flow with the original species. So now you have three species, this species, and this new species. And again, this is a major mechanism of speciation in plants because they tolerate these abnormal chromosome counts, but it is not a common mechanism in animals. Another way that we can get sympatric speciation is when organisms are living in the same area, but there's microenvironments within their area. So here's the broader area. So it's like a county or something like that. And then you have little areas of microenvironment that differ from each other. And so one example of this that we already talked about of a mixed environment is when you have, for example, heavy metal in some places and not in other places. And so that was causing grasses that were metal resistant to be living in these little patches and ones that were metal sensitive to be living in these little patches. And so even though they're living in the broader area, they're adapting to these microenvironments. And so you're getting potential gene flow blockage between these little areas. And so over time, you might expect that two different species would end up living in this area. Okay, because some are doing better on the metal patch and some are doing better on the non-metal patch. That you would get one ancestor population making two different species. Another example of this, and that's what this picture is of, is a bug that lives in the United States. And so in places like Ohio in the Midwest, you have areas where you have the native tree that the bug eats, and then you have areas that have the non-native tree, the apple. Okay, and so it's all in the same area, but there's patches where it's the native tree and patches where it's apple. Well, the apple is a really late fruit, so it typically is ripening in the fall. And the native tree is an earlier ripening. Bugs oftentimes have evolved to have timed their reproductive cycle to their fruit. And so this bug originally timed its reproduction to when the native plant is making ripe fruit. That's way its offspring will have an abundant food resource when around their reproduction. But in areas where you have apple trees, we're seeing selection where bugs that live where the apples are have a later timing of their reproduction. So the bugs that live with the apple trees are timing their reproduction later, and the bugs next door in the patch of native trees are having an earlier reproductive cycle. So again, they're still like hanging out in the same area. They're related to each other, and right now we consider them the same species. But over time, we might expect that this will diverge into two different species because, again, we're getting a blockage of gene flow between these two groups because their reproduction is being timed differently. Sexual selection is another way that we can block gene flow. And this gene flow blockage is based on female preferences. So you might have an original population of fish, but you have some females that have one preference for the male, and another set of females that have a different color preference. And so they're the same species, but some females like one type of male, and the other group of females likes another type of male. And so as the females of this group choose males of one color, and as the females of this group choose males of another color, that's blocking the gene flow. Right? So they're not going to be mixing up the genetics anymore. They're going to be going with the males of their choice, and they're going to be going with the males of their choice. And so over time, you'd expect that this would generate a new species, even though they're living in the same geographic area. So those 3 ways that you can get new species formation, even when organisms are living in the same geographic area.